and welcome to Instant Biology by Dr. Neela. The topic that we would deal today is Western blotting. Okay, so this is a very important technique in order to decide or in order to identify that whether a particular protein is present in your sample or not. So it is used to identify whether a particular protein is present in the sample that you are analyzing or not. Okay, so when I used to study these techniques, I had a very I used to forget the southern blotting, the northern blotting and western blotting. What are they used for and what do they identify? So I utilized a technique to learn these. So I would tell you in this course also. So when you learn these techniques, that is northern, southern and western blotting, what are they used for can be learned by a very easy two words. You can learn it by SNOW, snow, and DROP, snowdrop. If you learn these two words, then you can easily understand that which blotting technique is used for which macromolecule. Okay, so if you match S and D, then southern is used for DNA. Southern is used for DNA. If you use these two, that is N and R, then northern is used for RNA, and you can use the O in both of these. In both these words, O is present. You do not need to worry about that, and only last, that is W, can be matched with P, and W is for Western. W stands for Western and P stands for protein. So W stands for Western and P stands for protein. Okay, so with these two words you can learn that what is southern blotting, what is northern blotting and what is western blotting used for. But we in this particular lecture are not worried about southern and northern blotting. We are worried about western blotting. We need to learn that what is western blotting used for. Okay, and what are the steps of western blotting. So like other lectures, I have prepared a overview of this lecture, and we would be dealing with these points. Okay, so first of all, the western blotting is divided into 9 basic steps. The first step is sample preparation. Okay, so what do we mean by sample preparation? Sample preparation. So you know that a cell comprises of cellular components such as proteins. So in western blotting we are more focused upon, or we are worried about the protein component that is present inside the cell. Cell can be covered in case of eukaryotic cell, sorry, in case of animal cell it is covered by plasma membrane, and in case of plant cell it is covered by cell membrane and cell wall also. So you need to disrupt these membranes, you need to homogenize in some way. There are various methods that you can use in order to disrupt your cell outer covering, cell wall or cell membrane, and you can obtain the cellular portion, that is the cellular milieu, that is the cytoplasm or nucleus, the protein, wherever your protein is localized, you can go and obtain that particular region of your cell. So the first step is sample preparation. How do you prepare the sample? You first of all disrupt your cell, you disrupt the cell wall, disrupt the cell membrane, and then you obtain the protein component present inside the cell. So cell membrane or cell wall is disrupted, and after disruption what happens? You obtain it, and it can be disrupted through various methods such as sonication or mechanical shearing. You can use n number of methods, and after disruption you get your protein. You get your protein component. Now, what do you do for separation of protein? I have already told you in the earlier lectures. For separation of protein on the basis of molecular weight, a technique known as SDS-PAGE is very commonly used in most of the molecular biology labs. So in proteomics, SDS-PAGE is used in order to separate these proteins. So what do? You do SDS-PAGE. First was a sample preparation. We have prepared our sample, we have obtained our protein. Now we have to perform SDS-PAGE. So I would request you that if you have not seen my earlier lecture on SDS-PAGE, please go and watch that lecture and it would be clear to you. So in SDS-PAGE, what happens in SDS-PAGE? What happens? You cast a gel in which there are two types of basic gel. First one is the stacking. This one is stacking. I am just giving you an overview, and this one is the resolving. So what happens is your proteins here, the proteins that you had run on this gel are separated on the basis of their molecular weight. So whichever protein has a higher molecular weight, suppose there are three proteins A, B and C. Suppose there are three proteins. Protein A has a molecular weight of 60 kDa, protein B has a molecular weight of 70 kDa, and protein C has a molecular weight of 80 kDa. Then what will happen is that A, B and C, the C would travel the minimum distance because it has a higher molecular weight. It will travel the minimum distance. Suppose C would be present over here, and A, then it would be followed by B, and then it would be followed by A. A would travel the most distance. It will be like A, B and C. So this basically would come out of the SDS-PAGE gel. This we will get after the SDS-PAGE gel has run. We would have the proteins separated on the SDS-PAGE. So now a point comes that why we have already separated these proteins, and we have already know that our protein A, B and C are present on the SDS-PAGE gel. Now what is the use of Western blotting? You have already identified, haven't you, that your protein is present. So what is the need? Why do we need to go one step ahead to Western blotting? So we need to confirm that whether our particular protein of interest, suppose there might be another protein which has a molecular weight of 61 kDa, which cannot be differentiated from protein A with the help of SDS-PAGE. We want to be specific that our protein of interest is present in the sample or not. That is why Western blotting is used. Now Western blotting is a term that is used to that means basically to transfer what? Transfer proteins. In our case, transfer proteins, and transfer to where? Transfer on a membrane. What kind of membrane? A PVDF membrane or a nitrocellulose membrane. So two kinds of membrane can be used. This is point number 3. So we have already discussed about sample preparation. We have already discussed about SDS-PAGE. Now we are coming to membrane transfer. Okay. So let me write it down. We are coming to membrane transfer. Okay. So I have already told you that blotting means transfer. Blotting means transfer of our protein from the gel to the membrane. Which kind of membrane? You need to remember this. Which kind of membrane? Nitrocellulose membrane or PVDF membrane. So these two membranes are generally used. Why are these used? Because they have a strong affinity of towards the proteins. Proteins would go and bind to the nitrocellulose or PVDF membrane. Okay. So how does this happen, or how the proteins are transferred from the gel to these membranes? This need to be understood. Let us understand this by the help of a diagram. So how are the proteins transferred from the gel to this PVDF membrane? This we can understand this. So let us understand that suppose this is the negative terminal or the negative electrode. This Western blotting happens or is done in a Western blotting chamber or Western blotting apparatus. It has two electrodes. Okay. First is the negative electrode, then we will draw the positive electrode. So just after the negative electrodes, what you have a stack of filter papers. 2 or 3 filter papers are used. Then what do you have? Then you have your gel. Okay. So this is the SDS-PAGE gel. This is the side view. Remember, this is the side view. Suppose this was the gel that you had cast, and these are the proteins. Now you are watching in this way. This is the side view. Okay. So this is the gel. Remember, this is the gel. I'm also writing here over here. This is the gel. Okay. Now you have to keep in contact the PVDF membrane with this gel. Okay. Let me draw it with a different color. Okay, so this red one is the membrane. So the gel isn't just in contact with the membrane, and then again what you do is another set of filter papers you put just after the membrane, and then the positive electrode is present. This is the positive electrode. Okay, so now you know that electricity or electric current basically transfers from negative to the positive end. So what would be the flow of the current? It would be from the negative to the positive end. This is the flow of current. Okay, so what happens, what is the concept behind this, is that this entire chamber, this is inside a chamber. Remember, I have already told you that this happens inside the Western blotting apparatus, and this entire thing is poured, or this entire chamber is filled with a transfer buffer. Transfer buffer. Now what are the components of transfer buffer? Transfer buffer comprises of tris, it comprises of glycine, it comprises of methanol. Okay, so we would discuss the role of these, and let us first discuss that how are the proteins transferred from the gel to the membrane. Now these all are pressed onto each other. Okay, so when the pressure would be from upside to down, and negative and positive terminals are here, what will happen, that as and when the current would flow from the negative to the positive terminal, what will happen is that it will take the proteins along with it to the membrane. Okay, it will transfer all the proteins that were present onto the membrane. This would be facilitated by this transfer buffer, which would be filled entirely in this Western blotting apparatus. Okay, so one of the most important components of transfer buffer is methanol. It would facilitate the transfer of our proteins onto the gel, and it would also ensure that SDS you might remember that SDS is a detergent that was used to provide a unit negative charge on the protein that was used in the SDS-PAGE gel, and its purpose was also basically as a denaturing agent also it was used. So the purpose of methanol here is to restrict the transfer of SDS, that is the detergent, to the membrane, and only allow the transfer of proteins from the gel to the membrane. So this basic pattern or this sequence of the various components present inside the Western blot is very important. So this membrane is obtained. After this, what happens is you will obtain a membrane. Suppose, let me show you. After the Western blotting is done, you will obtain a membrane. This suppose is the PVDF membrane, and here this was the gel. Suppose here, here and here there are three proteins that you had to transfer on this. Now on the same position, these proteins would be now present on the PVDF or the nitrocellulose membrane. So what is the use of transferring this? What is the use of going through so much of a procedure, so much of pain? Why do we need to do this? This is just because we would be treating this membrane with various components such as primary antibodies or secondary antibodies, and you might be knowing that the gel is very fragile. If you keep on moving your gel, or you keep on washing your gel numerous times, keep on pouring different components or different reagents onto the gel, or transferring the gel from one container to the other container, that what will happen, that due to the fragility of the gel, it will break down, it will break, and you won't be able to continue your experiment. So what is this is the logic, that why do we need to transfer the proteins present on the gel to the nitrocellulose or PVDF membrane. So we have now transferred our protein to the gel to the nitrocellulose membrane. So membrane transfer is over. Now what we need to do is we need to block our membrane. Okay, so let me rub this out. So you have your PVDF membrane here. Okay, so now you have to go for blocking. So what does blocking mean? Blocking means to block all the non-specific sites on the PVDF membrane. What does non-specific site means? The sites where the proteins are not present, that can give false positive results. Suppose if in the upcoming steps you would be using primary and secondary antibodies. So the purpose of blocking is to ensure that your primary antibody does not bind to somewhere else, and it only binds to that particular protein against which it is made. Okay, so you have to block all this area. Suppose these are all the non-specific points, so you need to block all this area. Now what are the blocking agents? The most common blocking agent is BSA, bovine serum albumin. You use it, and there are other solutions also that you can use as blocking agents. Most important is bovine serum albumin. So when you use bovine serum albumin, it will block all the other regions, that is non-specific regions, and only the proteinaceous parts would be present. So the chances of your primary antibody that you would be using in the upcoming steps to bind to these non-specific regions would decrease to a substantial amount. So what do we do now? You have blocked your now you have ended your blocking step. Now you have to add primary antibody. So you know that antibodies, what is the function of antibodies? They are used to identify a particular antigen that has entered into our body. Okay, so these primary antibodies can be manufactured against a particular protein. Okay, so how can these primary antibodies be manufactured? If you want to identify this particular protein, this is protein B. Okay, if you want to identify that this particular protein B is present in our sample or not, then you have to design, or you need to get these antibodies against this protein B. Anti-B antibody, you need to get this antibody from the market, or you need to design it. Okay, so how can you design it? If suppose you have an animal facility and you are working on animals, you have mice as your working model. What do? You give this protein B to the mice. You give protein B. This is a very crude step that I am telling you. This needs a lot of preparation and a lot of ethical clearances. You need to give this protein B to the mice, and after some time what will happen is this protein B would be treated as an antigen by the mice, and then what will happen is the mice would prepare its antibodies against this protein B. So mice will produce antibody against B. Okay, against protein B, it will prepare those antibodies. So those antibodies we would use in order to identify that whether our protein B is present in the sample or not. Okay, so this would be followed by the pouring, or we will make use of the this antibody B. Suppose now we have mixed the antibody B and it has identified that this protein is B. This protein is B, and we have used this primary antibody. So this is the structure. I don't know if it is visible to you or not. I will just clear this and draw this once again. So this was the, suppose this was the protein that we were looking forward to. This was the protein and we are making antibody against it. So this is the primary antibody, same antibody that we have obtained from mice. Primary antibody can also be shown by 1 degree. Okay, so this primary antibody, what has it done? It has identified this protein B over here. Okay, so this primary antibody has identified this protein over here. Okay, so you would have mixed a lot of primary antibody. Now you want to clear off the antibodies that have not bound to protein B. So what would you do? You would go on to the next step, that would be washing. Washing would ensure that the non-specific binding or the antibodies that have not bound properly or that have bound to other regions would be washed away, and only the antibody that has bound to that particular protein or protein B would stay. Okay, so after the step of washing, what would you do? You would add secondary antibody. Now this is a very important step. What is secondary antibody? Secondary antibody is just like primary antibody, but the aim of that antibody would be to identify this primary antibody. Okay, so when you use the secondary antibody, it would identify this primary antibody. Okay, so this is the secondary antibody. This was the primary antibody, and the antibody that I have shown in blue is the secondary antibody. Its purpose would be to identify the primary antibody. Now, most often this secondary antibody is conjugated with an enzyme. Okay, it would be conjugated with an enzyme. It can be conjugated with an enzyme or a fluorescence molecule. It can be conjugated with an enzyme or a fluorescence molecule. What happens now? What is the use of this conjugation? This is done because when now you would use the substrate of this particular enzyme, or you would use a particular wavelength of light, then this would show light, or this would change a particular color, which would show you that our protein of interest, that is B, is present in our sample or not. Okay, now coming to washing would be the same. The reason for washing would be the same, that non-specific binding would be removed and only secondary antibodies that have bound with the primary antibody would be present over here. Okay, now coming to detection. Okay, now let us understand detection. So if enzyme is present in conjugation with the secondary antibody, now enzyme is present, and now you add your substrate. So what will happen? Product will be formed. This product can be a change of color or a light emission also. So if the color change is seen, now if suppose the color was blue initially and then the color changes to red, what will you see? What will you get to know? That our protein of interest was present. Only then the secondary antibody would be present, and secondary antibody, if present, would be bound to primary antibody, and primary antibody would only be present if protein B would be present. So it directly means if the color is seen, it would mean that the primary or the protein of interest, our protein of interest, is present. So the example that I can tell you about this is horseradish peroxidase enzyme is used, and it is the substrate is used for as a luminol. So it will give enhancement in the light. You can see a spot of light after this reaction occurs, and you can identify that your protein is present or not. So horseradish peroxidase is the enzyme that is conjugated with the secondary antibody, and luminol is the substrate that interacts with the horseradish peroxidase, and you can see an increase in the intensity of the light, and you can determine whether your protein of interest is present or not. So this basically is shown on the X-ray plate, because it is sensitive to such light changes, and you can easily identify that your protein of interest is present or not. So this was one of the examples that how you can identify this whether your protein of interest is present. So this Western blotting technique is used in a lot of instances, such as protein-protein interaction. And suppose you have a person that has been infected by some infectious disease or very serious disease, then what can you do? How can you use Western blotting for solving that case, or for telling whether the person has been infected by the same organism that you are thinking, or the disease is due to something else? What can you do? You can identify the protein present in that organism, and when if at all the person has been infected by that particular organism, then the proteins of that particular organism would be present inside that person. Okay, so you can design the primary antibody against the organism's protein, and if the primary antibody binds and secondary antibody binds, and then you get the change in the color or fluorescence, then what can you say? That yes, the particular individual has been infected by that particular organism, and the drugs can be then decided, and how the treatment regimen can then be decided. So this was all about Western blotting. If you have any queries regarding this lecture, you can post your comments in the comment box, and if you like my lecture, then please hit the like button and subscribe to my channel. Thank you so much. Have a great day.